Hello and welcome to the first experiment of the CBCH lab. The objective of this lab is to observe the formation of yeast spheroplasts after treatment with reducing agents and lidocase enzyme in order to analyze the spheroplasts later. But the reason why we are working with yeast is because the human genome contains counterparts to an estimated 1/3 of yeast genomes. These genes can be studied in yeast, and the findings can be applicable to human genes. This Venn diagram illustrates the counts of human yeast orthologs, or genes that overlap. Exposing the spheroplast is an important step in order to then extract the proteins or DNA you want to study. Lidocase, also known as zymolase, is produced by multiple bacteria, such as one known as Arthrobacter luteus. It degrades the yeast cell wall by hydrolase and glucanase activity. This solubilizes the beta-glucan within the yeast cell wall. To begin this experiment, the yeast first had to be cultured in liquid YPD medium for 2 days at 30 degrees Celsius. The yeast was then pelleted and washed with 10 milliliters of sterile water. The cells were then resuspended in a mixture of tris HCl and DTT, otherwise known as dithiothyretol, which reduces protein disulfide bonds that we will refer to as buffer 1. The cells were allowed to shake at 30 degrees Celsius for 25 minutes. The cells were then pelleted by centrifugation at 3000 G for 3 minutes at room temperature, followed by a removal of the supernatant. The cells were then resuspended in 20 mL of buffer 2, which consisted of sorbitol, potassium phosphate, and magnesium chloride. Sorbitol acts as osmotic support. It raises the osmolarity of the solution. The cells were then pelleted by centrifugation at 3000 G for 3 minutes at room temperature. The supernatant was then discarded. The pellet was then resuspended in 10 mL of buffer 3, which consisted of lidocase dissolved in buffer 2. The cells were then incubated for 30 minutes at 30 degrees Celsius while shaking. Here is how buffer 3 was made. It was made by adding 10 units of lidocase to 10 mL of buffer 2. Every 10 minutes, the cell suspension was checked for the presence of spheroplasts by placing 1 microliters of the suspension on a glass slide and by placing a cover slip on top. Furthermore, confocal microscopy images at 63x were also taken in order to view the cells in better detail than what you would get with a light microscope. After the spheroplasts were obtained, the last step was to wash the cells twice in buffer 2 in order to remove any excess lidocase enzyme. The supernatant was removed, and then the dry pellet was placed in -20 degrees Celsius for storage.